Hello, everyone. My name is Kenny McDurante. I'm a fourth-year veterinary student at the University of Illinois, and my research compared the effects of LH and EDTA on hematologic values in prairie rattlesnakes and Lake Erie water snakes. Anticoagulants are commonly used to prevent clotting in blood samples. However, samples placed in EDTA have been observed to result in hemolysis in various species such as eastern box turtles. Furthermore, studies comparing anticoagulants are currently limited in our snake species. The objective of this study was to evaluate the effects of LH and EDTA on hematologic values in prairie rattlesnakes and Lake Erie water snakes. We hypothesized that the absolute values of hematologic parameters would be different between species and anticoagulants, and that hemolysis would not be observed in any of our samples. We drew blood from the caudal tail vein in both the prairie rattlesnakes and Lake Erie water snakes and evenly divided our samples between one EDTA microtainer and one LH microtainer. Once we confirmed the absence of clots in our samples, we would spin them down in order to obtain our PCVs and total proteins. We additionally performed avian leukocyte white blood cell counts and 100 cell differential counts for each sample. After accounting for the effective species, the magnitude of the differences attributable to anticoagulant choice was relatively small, despite statistical significances. Ultimately, the clinical significance is negligible, as anticoagulant choice did not constantly or proportionally bias the values of any hematologic parameter, and all of our values fell within normal reference ranges for both species. Lastly, hemolysis was not observed in any of our samples. Ultimately, our results demonstrate that LH and EDTA are equally appropriate for use in prairie rattlesnakes and Lake Erie water snakes, as long as there is consistency with anticoagulant choice when comparing hematologic values over time or between animals. Thank you so much for listening.